Hi, good morning. It's Wednesday. This is the third, I think it is, the third lecture so far this semester. And we started out by talking about all of the different components, the different kinds of cells that are in the nervous system, and then the way the nervous system is organized. Then last time, we finally figured out how what is an action potential? What is a nerve impulse? And so then we had said, what happens at the end? So we had talked about all of these ions and how they're in all these different locations. Sodium ions are high in concentration on the extracellular fluid. Why? Because of the sodium-potassium pump, right? And there are leakage channels also, so it's kind of a messy process. The largest intracellular cation, positive ion, is potassium. It's on the inside, and we had said that this fluctuates in response to stimulation by other neurons. And we had said, what do? They secrete neurotransmitter substances. And then after that, we had said, the potential, I was blanking on the word, the potential within our neuron is going to be variable. We had said resting membrane potential was minus 70, but we had said we have all of these local / graded potentials, which are the fluctuations in the concentration of positives and negatives. That's what a potential is, right? When we say minus 70 millivolts is the resting membrane potential, what we're saying is we are slightly more negative on the inside than outside. And how much more negative? Minus 70 millivolts, thousandths of a volt. So we had said we have these local potentials that are fluctuating all over the place. They can be hyperpolarizing or depolarizing. And what does that mean? Hyperpolarizing means making it more negative. Depolarizing means making it less negative. And we had said we have a magic number, right? Threshold. And we had said threshold was minus 55 millivolts. So that means we've gone 15 millivolts from minus 70 up to minus 55. And and when we hit that magic number, what did we say happens? Action potential. And we had said there's a very specific location on the neuron where action potentials are generated. It's right at the base of the axon, and we had called it the axon hillock. And we said this is the trigger zone. And this trigger zone is where we're going to generate an action potential. And we said these action potentials are exactly the same. The steps in it, depolarization, repolarization, refractory, it's exactly the same. No greater, no smaller, as it passes down the axon. And we had said with depolarization, we're going to open a voltage-gated channel, and it opens in response to what? Minus 55 millivolts. Pops open, all those sodiums come into the axon, the door closes, and then we have another door open. I guess I should open it on this side, and all these potassiums go out. And so we end up with a flip-flop, sodium's in, potassium's out, and then it shifts down, sodium's in, potassium out, and it shifts down a little further. Sodium in, potassium out. And that passes all the way to the end of the axon. And what did we say happens then? You release neurotransmitter substances. And we'd said what happens? The neurotransmitter substance bubbles across the synapse, and it causes a change in the degraded potential in the next neuron, degraded or local potential. Okay, so today, I'm going to have to stop right now. We'll pick this up here in just a second.